Hi, everyone. Thanks for sticking around to the afternoon of the last day of PyCon to hear about my obscure use case. So as that wonderful introduction implicated, I'm an assistant professor at the University of Georgia, which can get awkward sometimes since my alma mater is Georgia Tech. And I have joint appointments with computer science and cell biology, so it's already within my job description what amounts to a mandate for very highly interdisciplinary research, one small slice of which I'll discuss with you today. So my talk is divided into three parts. The first part, I'll go over the background, some of the biology and kind of the clinical impetus for the work that I'm doing from a very high level. I'll try to keep things fairly basic. The next part, we'll talk about how we essentially generate numbers and statistics from the biology that we discussed in the previous part. And then at the very end, I'll go into what's the Python pipeline, actually connecting it with PyCon and the larger environment. So what are cilia? Cilia are microscopic hairs that line your throat, your nose, your lungs, your kidneys, your brain, lots of places. They protrude from cell boundaries, and they beat in these regular synchronous waves that you can see in this video here to move nutrients and trapped irritants where they need to go. And if you cut one open, it looks something like the structure you see on the right, on the bottom. Each cilium looks a little bit like the wheel of a bicycle, complete with spokes. The result of about 200 different proteins forming the structure. So the videos here are examples of what healthy ciliary motion looks like. If you watch the pattern closely, there's kind of a synchronous wave that just sort of moves along the entire boundary of the cell. There's obviously some variance to it. The one on the right doesn't look exactly like the one on the left, but in general, you can see kind of a coherent motion. So why do we care about cilia? Turns out they play a fairly critical role in keeping us healthy, and disrupting the regular synchronous motion of cilia can be indicative of a ciliopathy, a disease affecting the cilia directly. So, since cilia play an important role in delivery of nutrients and expulsion of irritants, disturbances in the motion can contribute to a host of conditions that can potentially result in lung scarring and permanent damage, necessitating transplants. And so to that end, accurately diagnosing ciliopathies is important. Cilia can also play an important role in embryonic development. It's been established in literature that embryonic nodes rely on cilia beating in circular motions to generate fluid flow in the early embryo, which generates some calcium signaling, which is all to say that, as the theory goes, it establishes the internal alignment of organs from the early embryonic stages. And so disrupting the proteins required to build that spoke and build the cilia and generate this motion and fluid flow can also result in a host of developmental defects, affecting the brain, affecting air-breathing mammals, correct formation of our hearts, correct placement of lungs, all sorts of horrible things. Congenital heart disease also shows a strong correlation with ciliary motion defects. And so simply put, this is all to say, we'd like to learn a little bit more about this motion and the role that it plays. And I guess, so on the top there is what, again, normal ciliary motion looks like. And in fact, the bottom one, there is cilia moving. You just can barely see it. It's highly, highly dyskinetic. So how do we diagnose ciliopathies? How do we diagnose abnormal motion? The current clinical methods rely on a combination of strategies, which I've shown here. First, is electron microscopy. You take a biopsy, stick it under an electron microscope, you look for abnormalities in the structure. Again, that bicycle and spokes structure that you saw earlier. Are some of the spokes missing? Is the wheel not fully formed? That sort of thing. This is very expensive, and ciliopathies can present without any discernible structural defects. Cilia can look fine under the electron microscope, but they're clearly not beating properly. The patient has lung scarring. So the second step is to measure the nasal nitric oxide levels. This is literally what's the concentration of nasal nitric oxide when somebody exhales. It's unknown why a higher NO concentration correlates with abnormal ciliary behavior, but while it's very cheap, it also results in a lot of false positives. So third, we measure the rate at which the cilia beat. So in some of those videos that I just showed you, you pick a pixel location, look at the signal waveform for that location over the course of the frames, measure the frequency, you get some beating frequency in hertz. And that kind of sort of correlates with normal abnormal behavior, but again, like nasal nitric oxide levels, not always. So again, more false positives. So finally, clinicians can examine live biopsies under a video microscope for motion abnormalities. This method is considered the most effective. You look at a biopsy under a microscope and say, yeah, they're not beating right. But it's done manually. That's how it's done. They look at it and they make a call. Depending on the clinician's training and bias, this can be very subjective with no relevance to studies done at other clinics. So doing kind of a cross-clinical examination is impossible. So then the goal of our project is to develop a method or framework which accepts a video or many, many, many videos, such as the ones you've seen earlier, and gives some quantitative description of the motion that we're seeing. So rather than arguing over the motion type, let's assign a number to what we're seeing. So we want to eliminate the subjectivity inherent in the current diagnostic process and replace it with a numerical description that is in some sense more objective. And then thereby defining kind of a rigorous definition of ciliary motion. So that was a 1,000-mile view of the biology and the public health impetus. So the core question for us, now we put on our computer vision hats, how do we quantify what we're seeing? How can we attach numbers to these motion phenotypes? So as we do often in computer science, we look for problems that we already know how to solve and see if we can cast our current problem in the mold of one of these known problems. There is a category of problems in computer vision known as dynamic textures. These are regular patterns of motion that have some amount of random noise to them, and you see them all the time. Clouds billowing, ripples in bodies of water, or as the example video is here, grass blowing in the wind. And when I play these two videos side by side, you can definitely, from some kind of qualitative sense, see similarities in the motion that we're observing. And so our thinking goes, we'll classify ciliary motion as a form of dynamic texture and thereby make use of all the tools in that area of research and just kind of see what we get. So analyzing dynamic textures, there are a few steps involved. We start with a video or region of interest, so over there on the left in step one, and compute the optical flow. Optical flow, for those who aren't experts in computer vision, it's a nice way of estimating the motion that happens between a successive pair of frames. Involves a lot of math and a lot of assumptions, but it gives you a pretty good overall instantaneous estimate. What you essentially get back for each pair of frames is a vector field, if you remember those from calculus. Each pixel is represented by a vector that points from where that pixel started in the first frame to where that pixel moved in the second frame. And you do this for every pair of pixels in every pair of frames. Oops. So that gives us the optical flow in step two. In step three, we compute the derivatives of the optical flow. How is the optical flow changing? And then finally, in step four, we can use the derivatives to compute what we refer to as elemental components. What are elemental components? They are things like scaling or zoom, so measure how much you're moving towards something or away from something. Deformation or biaxial shear, kind of how much it's being compressed or squeezed. And rotation, how much something is rotating. We picked these features because they're first order. You compute them directly from the optical flow derivatives. And they're kind of intuitive. I mean, rotation, you think it rotates, and that's exactly what it's computing. The unit is actually in radians per second. So in our studies, we found scaling was not particularly useful. If you think back to those videos, they're 2D. The cilia move within the plane of the video, so there's nothing really coming at you or away from you. But deformation and rotation were absolutely incredibly useful for our work. So what do these features look like? I mean, I kind of gave you an intuitive explanation of how we derive these, but in in the field, as it were, what do they look like? So I'll give you these two example videos. Again, very normal motion on the left, very abnormal on the right. And in each video, I've highlighted 3 points. The blue dot is a pixel very close to the cell wall. The red dot is a pixel towards the distal points of the cilia. And the black dot is background. It's just kind of a control to gauge ourselves against. And these graphs are the values of rotation that we computed at each of these pixels for each frame. So over 100 frames, what values of rotation did the blue pixel take, did the red pixel take, did the black pixel take, for a normal and an abnormal video. So this is kind of an example of extremes here. So it's a very normal sample and a very abnormal sample. But it paints a very clear picture regarding how these signals can vary depending on the phenotype. Rotation in particular differs by full order of magnitude between the samples on the left and on the right. And in particular on the left, we see kind of this nice periodic curve. So it rotates one direction and then goes back the other way. But in the abnormal one, we really don't see any signal at all. So this is kind of the quantitative form of our descriptors. And now we'll see how we can use that to differentiate rigorously between samples. So we'll go to, if anyone went to the Kalman filter talk yesterday, you've probably heard of this before, and I promise these are the 2/3 of the equations in this entire talk that I'll show you. We use something called autoregressive models, which involve the Kalman filter. The basic idea is this model invokes the Markov assumption that each observation is a function of some number of previous observations, and then we build a linear model from that. So with dynamic textures, we first project the original data, y, to a low-dimensional subspace. We do that with C, and then from there, we can model the evolution of this low-dimensional space using these parameters A1, A2, all the way through AD. So these are essentially our feature vectors. These model how the cilia move. So we compute these feature vectors for every region of interest or every video, and we use those to compare the motion that we observe. This is just kind of a visualization of what these what these matrices look like. Can't really tell anything obvious from this, but as a way of visualizing in a different way, we'll essentially simulate the motion that we would get from these certain parameters. So for example, the blue motion on the left is normal, the red motion on the right is abnormal, and all we're doing is we're picking, for a 3-dimensional subspace, we're picking a starting point at the origin, and we're using the transition matrices we computed for the 2 samples to essentially simulate motion. Given that starting point, we add the linear combination of the transition matrices with the next few points to discover, again, the whole Markov assumption thing. And we just run that for about 100 points, and these are the trajectories that we see, at least in the 2 top graphs. So you can very clearly see a difference between the 2 motion patterns. The normal blue has a very large variance and freedom to move in the subspace, while the abnormal motion, the red, by comparison, pretty much stays right around the origin. And when we fed these parameters into a classifier, we got about 93% accuracy. So we say, yay, we did pretty well. So that's a nice talk on cilia and on autoregressive dynamic texture modeling. Where does Python come in? I'm glad you asked. I'll throw open the door to the sausage factory here and show you how it's done. So the full pipeline has several phases to it. As in any data science project, the first step is collecting the data. In this case, we were collaborating with folks at the University of Pittsburgh Children's Hospital who had recruited hundreds of volunteers for their studies. They had obtained live cilia biopsies, recorded dozens of videos of the biopsies, but we didn't, and so they had all this data with them, but we didn't particularly care for the idea of passing DVDs and jump drives around, and who knows how we were going to identify regions of interest. So what we ended up doing was creating a website from Django, and just enough JavaScript to be dangerous, which would allow the researchers to upload the videos directly to our server and paint. You can kind of see the bottom left-hand corner of that right figure there. Paint little regions of interest on the videos directly in the browser. And then all these annotations would be saved into a SQLite database and use some nice SQL alchemy for object relational mapping. So that's how we collected the data. Once we had it, we needed to process it. These are large, uncompressed videos in AVI format. So we had to extract the regions of interest and convert them to data structures that Python could use. So for that, we used OpenCV, read the raw AVI files, pull out the regions of interest, and to do optical flow computations. OpenCV has wonderful optical flow implementations. We used the far and back algorithm, which was really fast, really efficient. Blew away the MATLAB algorithm that we were using in terms of performance. And then finally, to compute the derivatives of the optical flow and the rotation, deformation components, we used SciPy's not insignificant signal processing library. A huge selection of spatial filters and their derivatives for differentiating images. So with this whole entourage, this is how we had our rotation and deformation values. So once we had our rotation and deformation, we needed to compute the time series, and well, this was done by hand. PyCalman didn't exist yet, and I wasn't familiar with enough of the Python ecosystem to reliably search for alternatives, but at the same time, this was a fairly straightforward step. The pipeline, this is about half the pipeline right here, where we just took the raw data, did an SVD to get our low-dimensional subspace, and then from there, it was just a matter of a nasty-looking pseudo-inverse to get the transition matrices, but which was still only a few lines of code. So this step really used the SciPy linear algebra library, things like SVD, pseudo-inverse, all told about 10 lines of code, some serious indexing voodoo, but otherwise not too bad. So once we had our time series set up, then we went to the classification step. This was done almost exclusively in scikit-learn, except when we wanted to scale out the analysis to a much larger data set with hundreds of thousands of regions of interest. I mean, each individual video was about 2 gigs in size. So for most of the hyperparameter scans, we got away with using joblib to just scan the entire parameter space. But for some of the larger data sets, we throw it on a PySpark cluster. And then finally, we would use matplotlib and Seaborn for visualization, but we've recently started integrating its bouquet, right, into the aforementioned website to provide some feedback to users after they upload videos. We can compute some statistics on the fly and show them these nice interactive graphs right in the website. And that's essentially our pipeline. Okay, so there are 4 parts to my talk. Can't be a true academic without repeating myself several times. So here are some concluding remarks as well as things we'd like to explore moving forward. So our methods are good. We're recapitulating expert input. And ideally, we would like kind of what we designed here to function as a black box tool for clinicians. This was sort of the impetus. No clinician is also going to be trained or necessarily wants to be trained in computer vision. They would just like something they can feed their videos to and then get back the measurements that they're used to using. So they upload their videos, they get an analysis, and it's not a final call, but it functions as sort of another signal for them to make a final decision. And then the system provides for some kind of dialogue between the clinicians and the system itself, where clinicians can provide phenotype annotations, like they say, this looks normal, this looks abnormal. It's kind of a way of training it. And they can provide those regions of interest to help focus our algorithm on specific areas. So moving ahead, maybe the biggest thing we'd like to work on is throwing out everything we've done. No, not quite. But any clinician in this area will tell you the story is much more complicated than a simple normal-abnormal binary. Here are 4 videos of no fewer than 4 distinct motion patterns. Unfortunately, no clinician can agree on what all the distinct motion phenotypes are. Yeah, like I said, these are just 4 that represent kind of canonical motion subtypes, but then you get into motion mixtures, and then you ask a clinician from another clinic, geographically distinct from the first one, and you get 4 completely different answers, and so on. So we'll need to move beyond kind of the supervised framework into an unsupervised one that can discover these latent motion patterns. This is not only an interesting machine learning problem, but it has clinical relevance. There are a lot of studies coming out, especially in the last few years, that are showing potential links between ciliopathies and specific underlying physiological manifestations. Put another way, specific motion subtypes may correlate with specific physiological maladies, providing kind of yet another avenue for clinical diagnosis. And with that, I think I've left a few minutes for questions. Thank you for your attention. Thanks to PyCon for the opportunity to speak today. Thank you for the wonderful atmosphere and environment you've cultivated here. I've had an absolute blast this week. I'm happy to discuss this work or Python in general, or if you're a student or a postdoc, hiring you to help work on this. And with that, I'll take your questions.
So we are open for questions.
Hey,
Hi.
very interesting. We do something, my company does something very analogous, except for we do knee arthroplasty. I'm sure we could talk about it later, but our biggest challenge is, I don't know if you've even started to look at it, is getting FDA approval. Like, getting this to make sense is like one thing, and then you realize that you think you're done, and you've only done 10% of the work to actually create a product that could help people. Have you even thought about that or had any experience with that?
So kind of a bonus/limitation of collaborating with these folks at University of Pittsburgh Children's Hospital is, on one hand, it insulates us from having to worry about all that. They collect the data, they deal with the IRB, they deal with kind of all the clinical aspects. We just do the raw machine learning and computer vision. But on the other hand, it means we really don't have much of a say in how the work that we've done gets incorporated into any changes in the clinical diagnostic process. We're still not far enough along to really be able to make a strong argument that this should be part of kind of a new revamped clinical diagnostic process, but and so that hasn't really become an issue yet. But moving forward, yeah, I can see that we wouldn't be in the strongest position to kind of discuss with the FDA or any other approving body how best to incorporate this as sort of a, I mean, the way we're looking at it is it's just one more signal that clinicians would look at. Because right now they gotta go through all these other tests that I showed you, and then they make a decision based on their training. And ideally, we would kind of like this to behave that way, where the computer spits back a number or a series of numbers that says kind of this is where they fall on the spectrum, but you make the final decision. I don't know if that.
Yeah, we do that. Like,
Okay.
So yeah, just it's a lot of work just to be able to show a doctor a number. Just to get to, you can do that. It's a lot of
Yeah,
work.
but to a certain extent, we also want to try to, because there's this large amount of data between all the clinics, but none of the clinics can talk to each other, because that was kind of another problem I alluded to. Since there's no sort of quantitative objective definition of motion, you can't really compare studies coming out of multiple clinics. A good first start would be to develop at least a common language that everybody can use to start kind of comparing the results of their studies. And then maybe in the future we can get to something that's a bit more rigorous or publicly accepted in terms of diagnosing these things.
Thanks.
Hi, thanks for the very interesting talk. Well, I have a few questions. The first is there a baseline result? You know, you claim to have 97% classifications. I assume that is accuracy. And like, is there a baseline, like, you know, what is good and what is not good, right?
Yeah. Yes, so the astute listener will point out, wait, didn't you just argue that these identifications of normal, abnormal were subjective? Why are you basing the accuracy of your classifier on a subjective call? And I really don't have a good defense for that other than, well, we're trying to move to this unsupervised method. But you're absolutely right. Kind of the state of the art, I guess, is sort of that combination of diagnostic methods I showed you at the beginning. And one way that we compared it to a baseline was using that ciliary beat frequency method that I showed you, where you pick kind of a couple of pixels, you measure the temporal frequencies that are present at those pixels, and there's kind of a certain range, I think it's like 11 to 13 hertz, at which the ciliary motion is considered healthy. And anything below that or anything above that is considered unhealthy. And there are millions of extenuating circumstances that would say, oh, it's 5 hertz, but it's still healthy for XYZ. It's 15 hertz, but it's still healthy for ABC. But as a baseline, we kind of set that up and ran a classification, just, computing the frequencies, seeing if it fell within that range, calling it normal, abnormal, depending on that. And I think we got something like 65%.
Okay.
So less, but again, we made some simplifications. We didn't take electron microscopy into account. We didn't take nasal nitric oxide into account. So it's another problem in terms of just establishing a baseline, and we've been having trouble getting clinicians to accept this method just because of that. There's not really anything we can compare it to.
Yeah, so that brings me to the next questions about, have you thought of using an example of, like crowd compute or some sort of Amazon mechanical turks of a big group of doctors to classify this and, to serve as a baseline for your work? Because the last slide you mentioned that, a bunch of pictures and no one seems to agree on the same thing. So what does it imply? I mean, what does it mean, in a clinical sense?
Well, so in the clinical sense, at least in theory, there's a lot of evidence that says some of these motion patterns may correspond to specific diseases. So if we can plainly identify these motion patterns, then that might give us a really quick route to diagnosing a certain disease. So that's kind of the motivation. There are a couple of ways in which we're trying to address that. One is just plain and simple crowdsourcing. We had this idea where we throw up, a user visits a site, we throw up 2 videos and we ask the user to simply say, are these similar or are these dissimilar? And we kind of do that for enough videos with enough redundancy, and maybe that can start to give us an idea of kind of a consensus. Another way we're approaching it is now that we have these results, it got the attention of the clinical ciliary ciliopathy community. Not always the good kind of attention, but it definitely got their attention, and they've been wanting to kind of donate their data sets for which they have their own information, and seeing if we can kind of glom all of these together and start developing sort of a consensus. That's still somewhat, we have the data, but how exactly we're glomming everything together, especially when the recording frequencies for these videos can be different, which can completely negate the effectiveness of these time series models if the sampling frequencies are all completely disparate. But there are some other thoughts that we have as to how we might be able to start comparing these. So, short answer, there are a lot of ideas that we're working with, but no straight answer just yet.
Thanks.
I noticed from at least one of the slides that you were going from nasal biopsies. Mm-hmm. How does this translate to other cell types?
That is a very good question, and one which we've only just started scratching the surface of. So I mentioned that cilia are all over all different cell types in your body. Yes, this study focused exclusively on nasal biopsies, but we've been seeing very similar things in rat brain biopsies. And so we've actually started looking at some of the ciliary motion patterns in brain cilia, and we're finding that although the specific global parameters of that motion may be different, kind of the relative variations are holding holding steady. Like the cilia beat a lot faster, we're finding, for instance. And we have to kind of take that into account, but otherwise we're still, the same machinery seems to work pretty well in that case. I don't want to go too far in that area because we haven't really done a whole lot of work there yet, but our hope is to be able to apply the same machinery to cilia in lots of different contexts.
Another thing that I've realized is that from your discussion, you're making some assumptions about the data and what to look for. And recently, especially in the news, there's been a lot of talk about deep learning and the ability of such pipelines to take raw video, in effect, and derive their own sense of what's important and what's not important. Have you considered looking at that line?
Very much. In fact, I keep toying with this idea of designing a convolutional recurrent net to look at these videos and try to come up with sort of what's important on their own. That is something that honestly, if I could recruit somebody here to help me work on, that would be my ideal, because frankly, I just haven't had time to go that route yet. But that is indeed actually the kind of the secret direction that I've been wanting to head for quite a while now.
Thank you.
All right.
So thank you, Shannon.
Thank you very much.